In this video, we're going to really start to get down and take a look at the idea that membranes are what we call selectively permeable. Permeable means things can pass through, but selective means that only some things can pass through. Membranes are selectively permeable because they have that amphipathic nature, a hydrophilic head and a hydrophobic core, which are at direct odds with one another. So things that can associate with one side of the membrane cannot readily pass through that hydrophobic core. We're going to take a look at two types of transport, passive and active transport. And then we're going to talk about different ways in which the cell can achieve these. Make note, although it's easy to think about passive and active transport simply by taking a look at energy, the truth is that the way in which passive and active transport are separated is due to directionality. There's only one orientation for passive transport to occur, and that is from areas of high concentration to low, while active transport will always be in the reverse. Diffusion is the random movement of molecules, and this can include across a membrane. So passive diffusion is going to allow small molecules like oxygen, which is going to be a small nonpolar gas, to be able to pass through the membrane directly. Only some substances can do this, and because it's a form of passive transport, they can only go from areas of high concentration to low. This diffusion down the concentration gradient requires no energy, and it's because the cell is attempting to be able to even out distribution on either side. Diffusion doesn't stop when the two sides are equal, but it will maintain an equilibrium where the two sides, even though they continue to cross, even though the substances continue to move, will always move at an equal rate across either side. Osmosis is the word that we use to describe the diffusion of water. And water will always diffuse from a region of low solute concentration to the region of high, because low solute concentrations have more free water than high solute concentrations, which have less. And so the water will move from areas of high water concentration to less. So now I want you to think back on our previous lecture about the properties of water, and pause the video here to think about this. What is the reason that there are less free water molecules in higher solute concentrations? In order to understand the answer to this question, you need to understand how water interacts with solutes. Because of the nature of water and its ability to hydrogen bond, it's going to form hydration shells. Now, water that is being taken up by the formation of hydration shells is no longer free to move. And so areas that have low concentrations of solute have more water that's free to move. Areas with high concentrations of solute have more water being taken up with the formation of these hydration shells. When we take a look at these properties of water, the ability for osmosis to move water across a membrane, it's important to consider relative concentrations. Well, when solutions are the same, solute concentrations are the same across a membrane, we refer to these as an isotonic solution, where iso means same. And while water will continue to move across the plasma membrane, there will be no net change in the concentration. When solute concentration is greater outside than inside the cell, this is known as a hypertonic solution. The cell will lose water because it has an excess of free water. But when the solution is hypotonic, then the concentration is less outside, and so water will flush into the cell. If you were to take a look at unwalled cells, like our red blood cells, we find that in a hypotonic solution, water rushes into the cell and it will burst the cell in a process known as lysis. This is also known as osmotic lysis. If you have a hypertonic solution, water will rush out of the cell and become shriveled. In a cell that has a cell wall, the same properties of osmotic pressure apply, but the cell's response will be a little bit different because cell walls can protect the cell against that osmotic lysis. And so plant cells, for example, an example of a eukaryotic walled cell, actually want to be under hypotonic pressure. This actually helps to maintain proper pressure in the cell, known as turgidity. And that's why when you see plants that have had lots of water added to them, they actually look plump and green and pleasantly ripe. Non-walled cells have to use other mechanisms to regulate osmotic pressure, known as osmotic regulation. What I'd like you to do is take the next 3 Socrative questions to test yourself, to see if you can understand how non-walled cells, like ourselves, regulate osmotic pressure. So pause the video here and do the next 3 questions. All right, so before we start, let's look at one example of how non-walled cells, like this paramecium, can control osmotic pressure. Well, paramecium have a special organelle known as a contractile vacuole. It's a specialized vacuole that can push water out of the cell. So when the cell is surrounded by an environment that would be hypotonic, so a hypotonic solution, that means water will rush into the cell, and if it does nothing, the cell will undergo osmotic lysis. But a contractile vacuole can push that free water back out of the cell, allowing the cell to osmoregulate. For an algal cell that lives in the ocean, well, the ocean is salty. This would be a hypertonic solution, and so the water is going to be then flushing out of the cell into the surrounding environment, and the cell is going to become shriveled. If you contrast this with human cells osmoregulate through the control of solutes. So the paramecium that we discussed osmoregulated by moving water, but our cells don't have contractile vacuoles. They regulate by pumping solutes across their membrane, ions being pumped out, which we're going to talk about in a moment. If a human cell was swollen, it could relieve the pressure by transporting ions from inside of the cell out. By doing so, some of that free water will be taken up in the formation of the hydrogen cells from these solutes, which were now pushed out. In order for cells to be able to do this, we need to make use of proteins. So proteins, when they participate in the process of transport, are known as facilitated diffusion or transport proteins. So when we talk about facilitated diffusion, we're still moving down a concentration gradient. So from an area of more concentration to an area of lower concentration. These transport proteins simply speed up the process by which molecules move across that membrane. Active transport is the use of proteins with the addition of energy to move in the other direction. Remember, the difference between active and passive transport is the direction in which the solute travels. In facilitated diffusion, transport proteins are going to move molecules down their concentration gradient. They're just going to do so faster than if we relied on passive diffusion alone. In active transport, we're going to move them with their against their concentration gradient, using the energy from the hydrolysis of ATP.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Not only does the transport of ions across the eukaryotic plasma membrane allow the cell to osmoregulate, but ions have a charge to them. And so this allows the cell to create this tremendous force known as an electrochemical gradient. Now, this is going to be a theme. Electrochemical gradients are a big deal when we talk about how cells do work and achieve metabolic activity. So please pay close attention. An electrochemical gradient is a type of potential force whereby there's a difference across the membrane. This is a dual force. The fact that the ion is of a greater concentration on one side of the membrane than the other is a chemical force. But because ions are charged, there's an electrical force where there's a greater or a different charge on one side of the membrane than the other. So the electrochemical gradient is such a potent potential force because it's the sum of two potential charges. Some ion transport proteins can generate that voltage across that membrane. Again, remember that these ions have a charge to them. And so a proton pump can actively pump and transport hydrogen ions out of the cell to help achieve this. So these protons, hydrogen ions are also known as protons, have that positive charge which creates a differential across the membrane. These pumps help to store potential energy that the cell can use to do work because they've created this electrochemical gradient. We can also harness this electrochemical gradient's potential energy by doing something known as co-transport. When we utilize energy to pump these protons across the membrane, we create the electrochemical gradient. The potential energy stored in that electrochemical gradient allows that proton to diffuse back down through a protein. So this transfer down this protein is going to be a passive form of transfer, going from an area of high energy to low. But because this passive transport results in the release of energy, we can couple it to the transport of another molecule. When we do so, the energy used to pump the proton gets harvested in the transport of other substances. This is co-transport, co meaning both, because both of these substances will be pumped back across. This is a form of active transport because you must first provide energy to pump the protons, creating the electrochemical gradient. So you wouldn't have this process if you didn't provide energy to begin with. Energy like this can be used to transport large molecules across the membrane. They can be transported out of the cell, so this is known as exocytosis, transporting things out of the cell, or endocytosis, endo meaning internal, because we're taking substances from the outside of the cell and bringing them internal. So exocytosis and endocytosis both rely on transport across the membrane through the use of energy. Now, when we transport things into the membrane, this could include, for example, the production of a food vacuole. So when the cell uses endocytosis to bring in a food particle, which one of the organelles that we've learned about can merge with that endosome, another word for an endocytic vesicle, that aids in the digestion of these food particles? Tell me in Socrative, which one? Good, this is a lysosome. Remember that lysosomes have in them this process of hydrolytic enzymes, so they can do a hydrolytic digest, which breaks down macromolecules, which is really all that food is. right? Food is components of macromolecules that have simply been obtained from another source. So now what I'd like you guys to do is to take a look in your textbook, make sure you emphasize and look closely not only at the macromolecular composition of membranes and how those macromolecules in the membrane provide the structures which give the functions, but also look more closely at protein functions, because the embedding of different proteins in the membrane gives different functions to the membrane. You'll also be asked to identify, based on situations, what the actual conditions are for the transport of water across membranes. And then I'll ask you to contrast that osmotic transport with other kinds of transport across the membrane. How can we distinguish between passive and active transport, for example? And by what mechanisms can transport proteins aid in the transport of molecules across the membrane? Finally, be sure that you understand how active transport can be used to generate not only a gradient, but an electrochemical gradient. And that electrochemical gradient can be used by the cell to do work. Once you've gone through and reviewed these processes, take a look at this video, which is going to be posted on underneath of this one, for a more visual view of these and a review of transport across the membrane.